Okay, so if you remember, I'm not going to pull up the slides yet because I don't want the screen kind of blanking out what's on the board. So what we ended with on Wednesday was talking about why surface area to volume ratio matters, right? And I realized maybe people are not as comfortable with ratios and math, but it's actually, I like this worksheet because it breaks it down really simply. So all you have to do is simple multiplication and simple division to figure out how efficient a cell is. So the whole goal of learning this idea of surface area to volume ratio is to figure out how efficient a cell is. And when you think about a cell, I guess I'll draw, we're going to imagine a cubical cell because it's a lot easier to deal with the math of a cube than it is a sphere. So that's why we're doing cubes. So we're going to imagine just kind of differently sized cubes, basically. So really small versus, you know, in between versus big. So we're talking different size cells, different cubes, and we're wondering which of those is more efficient. Is it better to be big or is it better to be small? And this is all from a the perspective of what can make it into and out of the cell. So the cell needs a lot of stuff to come in and it needs a lot of stuff to go out. The next chapter is basically all about that. So it's about the plasma membrane, talking about movement of materials in and out of the cell, which is happening constantly in every single one of your cells in your body. So the amount of surface area there is versus how much volume needs to be supported inside of that surface area is why this concept is important. So hopefully that sort of makes sense, but we'll run through this math, and then I think the questions at the bottom kind of do a good job of bringing all that together when it's just a bunch of numbers. So for, let's see, cube 1, we had a side length of 1. Well, never mind, that's all done. So cube 1 was already completed, right? So we can see that the surface area was 6, and that's going to be centimeters squared. So area is squared, and then volume is cubed. That's the whole underlying mathematical reason for why the surface area to volume ratio matters, which hopefully you saw by doing that math. All right, so for cube, it's kind of in the way. So we did cube 2, right? And what did we get? We'll just go to the very end there. What was the efficiency of that cell? 3, yeah. So that's the efficiency of the cell. For cube 3, that basically means 3, the side is 3 centimeters in length, so each side. So to get the surface area, what did you guys multiply? Oh, okay. Yeah, so for the side length, you're going to do, for the surface area, 6 times 3 times 3, right? Does that make sense? So if you look at the top, the surface area, and you guys on Zoom, hopefully you can see this, the equation for surface area up here, 6, because there's 6 sides on a cube. So 6 times length times width, basically. That's going to give you how much area there is on the outside of the cube. So, actually, I'm going to set this up a little bit better. So surface area, and we'll have volume, and then we'll have ratio, and then we'll have efficiency. You guys see all of that? There we go. Yeah, this is great. Okay. So for cube 3, that means each side is 3 centimeters long. So you would do 6 times 3 times 3, right? What does that come out to? What was it? 54, yeah. So 54 centimeters squared. And then the volume is simply just each side multiplied by itself. So 3 times 3 times 3, which is what? Right. So 3 times 3 times 3. That's 27 cubic centimeters. So that ratio, and I said reducing the ratio can kind of help you understand things, but let's just keep it simple and not even reduce the ratio. So for those of you who aren't all that familiar with ratios, all it is division. So for the ratio, you can just write 54 to 27. So this is the SA to volume ratio. So 54 colon is usually the 2 in a ratio, 54 to 27. And for the efficiency, all you do is divide those. So 54 divided by 27, which comes out to what? 2, yeah. Does that make sense to everyone? Okay. So cube 4. What did you guys get for surface area for that? 96, yep. So that's going to be 6 times 4 times 4, right? Because now the side length is 4. So 96 centimeters squared. And the volume would be 4 times 4 times 4, which is 64. 64 centimeters cubed. So that gives us a ratio of 96 to 64. 96 divided by 64 is, yeah, 1.5. 96 over 64 equals 1.5. Okay, so we went from, what was the efficiency of the first cell, which was done for you, was 6, right? Then we made it a little bit bigger and the efficiency dropped down to 3. We made it a little bit bigger for cube 3 and it dropped down to 2. Then we made it bigger and the efficiency dropped down to 1.5. So this pattern is going to hold. The bigger a cell or anything becomes, the less of, well, yeah, cell. The bigger a cell becomes, the less efficient it's going to be. So that's why there's an upper limit to cell size. We can't have just 5 big cells in our body. Basically, we have to have a lot of little ones because otherwise they're not efficient at all. Does that make sense? Okay. There's just too much volume inside of a cell this size compared to how much surface area there is to allow materials in and out. So the questions at the bottom, which cube had, I think the first 2 were really repetitive. Which cube had a larger surface area to volume ratio, 1 or 4? 1, yeah. So I don't have it up here. Cube 1 has a larger surface area to volume ratio. It's the same thing as the efficiency of a cell. So if you look at number 2, for our purposes, you don't really need to know much more about ratios. I'm not worried about the mathematical aspect. So if you can understand how to get to the efficiency, that's what I'll ask you about. And I will ask you about this on an exam. So thinking about the efficiency of the cell, you just want to know you'll need the surface area divided by the volume. That'll give you the efficiency. So you can kind of compare this relative efficiency of one cell to another. So those first 2 are, cube 1 has the greater surface area to volume ratio, which means it has a greater efficiency. So you do want to know, hopefully you guys can see this okay. The SA to volume ratio essentially equals the efficiency of a cell. So as this increases, the surface area to volume ratio, this increases. So they're directly related. As surface area to volume ratio goes up, the efficiency of a cell goes up. Let me make sure you guys on Zoom can see that. Sorry, the lights are kind of reflecting a little bit. So hopefully you can see that okay. So that's the take-home message of all of that. And that was just one slide in that PowerPoint. So I know it probably seems like it's not that important, but it is a really important concept in how basically every animal, including ourselves, can actually function. So the last two questions, what type of cell, smaller or larger, would have better efficiency? Smaller, yeah, so same idea. It's kind of repetitive, but hopefully that just brings the point home. A smaller cell is going to be a lot more efficient. There's a lot more surface area compared to how much volume there is. And then the last question, so 9 small cells, we'll say they have the same volume as 1 large cell. So which has more cell membrane for nutrients and waste to pass through? The 1 large or 9 small? 9 small, right? Because you have 6 sides for the amount of volume versus, so 6 sides versus 6 times 9 sides, essentially. So if you have 9 small cubes, you have a lot more surface area versus 1 big cube. So again, that's why our tissues and everything are made up of a lot of really tiny cells. They're just a lot more efficient that way. Okay, any questions about that concept or that handout? Anyone on Zoom? Okay, awesome. So yes, I will ask you about that on the exam. There'll be something about that concept on the exam, so make sure you understand that well. Okay, so next, I'm actually not going to lecture a whole lot today. I'm going to have you guys do an activity. We're kind of going to sort of switch pages a little bit here. We're still going to talk about the cell, but not like a mathematical version of the cell. We're going to look at the different organelles in a cell. And I'm going to assign an organelle to, I'm going to break you guys into 7 groups across Zoom and in person. And I'm going to assign 1 organelle to each of the groups. And what you guys are going to do is basically make an analogy for that organelle. Say, what did I use as the example? So the cytoskeleton is kind of an organelle. It's part of the cell inside, and it helps structure the cell and move stuff around. So you can think of a cell as a really, really tiny city in a way. It has energy production plants. It has kind of a city hall that directs everything. It has waste treatment plants. It has a transportation system. So the cytoskeleton, because it moves stuff all around, you can think of sort of as like a subway or railroad tracks or something like that in a city. So what you guys are going to do when I break you up into groups is take the organelle that I've assigned you, make some kind of analogy to a city. It can be a totally made up city, but the function of whatever entity of the city you think best represents that organelle is what you want to come up with. So base it on the function of the organelle. So I'll hand you some slides that describe what each of the organelles do. So you can find your organelle, figure out what it does, and then make an analogy to some part of the city. And it doesn't have to be perfect. This is really just kind of a helpful way to remember the organelles, and it's much better than me just boringly reading through the functions of organelles, which gets really, really boring. So then you're going to present it to the class. So you'll take about 5 minutes to do this once I get everyone set up, and then explain why you chose that part of the city to represent that organelle. So you guys on Zoom, I have put up a couple of different handouts that I'm going to give to the people in class, and I'm about to break you up, so hopefully you're paying attention when I put you into the breakout groups. So you're going to download this cell organelle activity. It should look something like this. And then also there are cell organelle slides, which is just a PDF that looks like this. These are the same slides that are at the end of the PowerPoint lecture on the cell theory and structure. All right. So I'm going to while you guys are getting those downloaded on Zoom, I'm going to hand these out to the people here in person, and then I'll come back and put you guys in groups and tell you which organelle you're assigned. See ya. Okay. And all of the organelles are on here, so Definitely take notes when people are presenting their Organelle. And I'll give you guys in just a second. I'm going to group the Zoom people up first so I can figure out how many more groups we need in here, but probably 1, 2, 3. Groups. All right, for those of you on Zoom, I'm going to put you in breakout rooms and then look for a message from me, and I'll just say your names and which organelle you're assigned. Okay, 4, 7, perfect. All right, so if you guys want to group up, I guess by table more or less. So if you two want to be in a group, and then you two can be in a group, and you two. And let's see. We'll start with, so you guys can be the cytoskeleton. And then second table back, you guys can be the nucleus. And then endoplasmic reticulum. Yes.
Okay, so you were saying like basically take a part of the city
Yeah.
and describe it as our organ. Right?
Yeah. Yeah. Shay and Sophia, can you join your breakout rooms, please? Or your breakout room? You're in the same one. Okay. Okay. No pressure. All right, so cytoskeleton, do you guys want to go first?
Okay, so we did subway because a lot of cities are built around transportation, so that helps provide structure and keeps the city moving because it gets workers to and from where they need to go.
Nice, yeah, that was a really good explanation. So they said they compared the cytoskeleton to a subway, and that's because it helps move people from where they need to go and helps provide some kind of structure and link between different parts of the city. And that's a lot of what the cytoskeleton does. So the slide I have up here has the info on the cytoskeleton. So this is from, well, it's on the organelle handout, but it's also in that PowerPoint lecture for cell theory and structure. So it's gonna do a lot of things, kind of determine cell shape, support the structure, move things around within the cell. Those are the main functions of the cytoskeleton. It is a skeleton, it provides structure as well as moves things from one place to another. There's a few different filaments and fibers here. You don't need to know them. That's mostly just for your own information. So there are different pieces. It's not all just one kind of thing, but don't worry too much about the differences between those. Okay, cytoskeleton and nucleus. What did you guys come up with for nucleus?
We chose a mayor.
Okay.
It's like the nucleus is the main organelle. Every cell has one nucleus. Every county has one mayor.
Yeah.
City. Every city has one mayor. And it controls a lot of the things that's going on in the city, like the nucleus does.
Exactly. That's perfect. Yeah. So they said that the nucleus was like the mayor of a city, which is a perfect analogy. The mayor basically tells everyone more or less what to do. I guess it would be more of like a fascist kind of mayor, because the nucleus is really in charge of everything. There's no like voting by the constituents of the cell as to what to do. But yeah, the mayor is in charge. The nucleus is totally in charge of the cell, determining what functions, what's being made, what's being kicked out, all of that kind of stuff. So it's the central deciding entity of the cell. Exactly. Good. So what else? Yeah, it is the largest organelle. For the most part, cells have one nucleus. And an important aspect of the nucleus, and we'll get into this when we talk about chromosomes and stuff, is that this is where the hereditary information or genetic information is stored. So all of your DNA is in your, well, most of your DNA is in your nucleus. So the important DNA that kind of makes you who you are is in your nucleus. Okay. And then next we have a picture of the nucleus. I forgot to put this up here. So this kind of gives you an overview of different aspects of the nucleus. There are pores on the nucleus. So hopefully you guys on Zoom can see that. There's these pores where material goes in and out. So the nucleus can still move stuff in and out of itself as well. Okay, ribosomes. You guys are ribosomes, right? Oh, whoops, that's right. I have this in a different order. There we go. Okay, you guys are ER.
So we said it was like a hospital or a pharmacy because what it does is it produces proteins, phospholipids, and steroids, and helps by detoxifying like drugs and alcohol. And kind of like hospitals and pharmacies, they produce things to help people feel better.
Yeah. That's a great analogy. So they said that the ER, the endoplasmic reticulum, was like a hospital or pharmacy because they're making things that detoxify a lot of other things, basically. So they're kind of making these helpful drugs and kind of helping to heal a lot of the cell. So that's perfect. The endoplasmic reticulum is, it has two parts technically. They're pretty similar. They're all connected. The important aspect here is that the rough ER, basically, rough ER and smooth ER are the two different parts of it. So the rough ER is just rough because it has these ribosomes on the outside. And we'll do ribosomes next, I think, right? Yeah. So yeah, ribosomes are going to come next, but they make proteins, essentially. Sorry, I didn't mean to get into someone's explanation. But yeah, so the rough ER is going to produce some proteins, and then the smooth ER doesn't have ribosomes, so it's smooth on the outside, but it's still all connected. It's all these channels, so they're all connected with each other. Makes phospholipids and steroids, and like you said, detoxifies alcohol and other drugs. All right, so that is the ER, or endoplasmic reticulum. So now we're going to the first group on Zoom, room 1. That was Shay, Sophia, and Walker. You guys did ribosomes. So what'd you come up with for ribosomes?
We basically said that it was like the courthouse that hold all the information, like laws. So it was the courthouse for us.
The courthouse that holds all the information?
Yes, ma'am.
Oh, okay. Like reading the genetically coded messages. Okay, yeah, that's a good. I haven't heard that before. I like it. Ribosomes are kind of tough for me to figure out how to make an analogy in a city. So yeah, they read coded genetic messages, and the courthouse basically holds on to, yeah, all of the records for a city. That's perfect. I love it. And I haven't heard that before. So good job. So yeah, they are going to take genetic messages and basically make proteins from those genetic messages. That's the whole point of ribosomes. They make proteins. They make proteins using this genetic information. And we will talk all about that when we get into a much later chapter about how that's done. So you'll see this again, and we'll learn how this amino acid chain is made using this specific genetic code. But this is the ribosome, this kind of bluish thing on the back. That's what's actually reading the information. Yeah, I think that's the most important part. Ribosomes make proteins. All right, good. So we did ER. The Golgi apparatus. That was Anna, Chloe, and Danny, right? You guys did Golgi?
Yes, we said it was like a post office or like a packaging plant because it packages, processes, ships, like stores and like receives all kinds of stuff. And so we chose it because like post offices package and ship, and so do packaging plants.
Yep, that is a perfect analogy. And I think honestly, in most textbooks, that's what is used as an analogy for the Golgi. So it's the post office, if you guys didn't hear, sorry. So she said it was like the post office because it packages and ships materials. Can you open the door for her in the back? Sorry. Thank you. So yeah, the Golgi apparatus is really important in kind of putting a few finishing touches on proteins and then getting them all packaged, getting them ready to ship out where they need to go. And they get shipped through what are called Golgi vesicles. So this is kind of what the Golgi looks like, these kind of stacked, flattened sacks. And coming off, when a protein is ready to go, one of those Golgi vesicles will have a protein inside of it and basically butt off of the Golgi body and then go to where it needs to go. It might go to another part of the cell. It might go to the plasma membrane to be excreted from the cell and used elsewhere for another cell. So yeah, packaging, processing, shipping, that's the Golgi. And there's actually a song that talks about the Golgi apparatus, which is funny, or the Golgi, I guess, yeah, by Fish, if you guys know the band Fish. They talk about Golgi. Okay, so we have two more now. Next are the lysosomes and peroxisomes I put together. So that was Jamera and Jeremy. What did you guys make an analogy for the lysosomes and peroxisomes?
We said that they were like the garbage trucks because they get rid of the molecules that aren't supposed to be in the cell.
Yeah.
Yes, because they're breaking down.
Exactly, yes. So the lysosomes and peroxisomes, they're really similar. So they said they were like the garbage trucks. They're taking waste and getting rid of it. They are basically little balls of enzymes that break down waste. Yeah, that's a perfect analogy. So a couple other important aspects I want to mention. Lysosomes in particular are responsible for digesting a cell's extra organelles. So sometimes a cell doesn't really need as many ribosomes or maybe as many Golgi vesicles as it did at one point. So lysosomes will basically eat those and break them down. And I'm not quite sure what the process is for this, how it eats itself, but lysosomes will digest the entire cell if the cell needs to die. So it will somehow ingest everything around it and the cell will then disappear. I don't know the final step of that, like where the lysosome goes, but that's called autolysis, which is cell suicide. So digestion of a surplus cell by itself, which is kind of crazy, but that's what the lysosomes do. In our immune system, there's actually a really important cell, type of white blood cell called a macrophage, and they have a bunch of lysosomes. Those lysosomes in these immune cells are basically eating bacteria that are invading our body, or viruses or protists or anything that's coming in and trying to invade us. Our immune system has a lot of lysosomes. Our immune cells have a lot of lysosomes to take care of all of those kind of constantly invading entities into our body. Oh yeah, I did want to mention really quick, I know we'll get to, sorry, mitochondria and chloroplast guys, we'll get to you in just a second, I promise. I wanted to mention a specific disease. It's actually a lysosomal storage disorder. So this is where an individual's lysosomes basically can't break down lipids or fats, and it's called Tay-Sachs disease. So the lipid digesting enzyme specifically is missing from lysosomes. It can break down other things like carbohydrates and organelles, but it can't break down lipids. And those lipids eventually build up and hinder the brain from creating neural connections. So the brain is impaired by accumulation of these lipids. It is genetic, so they know about like where the mutation in the DNA is found. It's really rare, but it does end up leading to death within about 4 years of when a child is born. So it starts to show up around ages 3 to 6 months, where children basically can't roll over or sit up or do any of that because their brain is basically impaired by a lot of lipid buildup. And yeah, they rarely live beyond 4 years. So it's a really serious disease and caused by something that we wouldn't necessarily think of as causing some kind of major childhood disease. So Tay-Sachs disease, just a kind of mutated genetic disorder dealing with lysosomes. Peroxisomes, basically the same as lysosomes. They just break down different things. So I often group these together. They're going to detoxify a lot of alcohol and other drugs, in addition to, well, where the lysosomes kind of do, they break down the biological macromolecules. Peroxisomes are really important in detoxifying any alcohol or drugs in your system. And those reactions, they're called peroxisomes because they make hydrogen peroxide, which is actually really toxic. So you don't want to drink hydrogen peroxide if you were ever considering it. It's toxic, but they then break it down from hydrogen peroxide into water and oxygen, essentially. So they break it down into actually fairly useful products for our cells. So that's peroxisomes, also kind of a waste treatment facility for a cell or a garbage truck. And then finally, these are really important ones, so I hope you guys came up with a good one. The mitochondria and chloroplasts. I said you could kind of focus on the mitochondria if that was easier. So, yep, Brandon and John. Oh.
Yeah. Oh.
You got it. You did the chloroplast. I did both, so I'll do chloroplast. You got mitochondria.
Yeah. So basically for the mitochondria, it would be just like the energy plant of the city, since it supplies all of the energy. It makes energy and power for the cell, basically.
Right, yeah, exactly. So he said the mitochondria are basically the energy plants for the city. They're making all of the energy, the ATP. Mitochondria are the little energy centers.
And then for chloroplast, I did like a farm, because you know how in plants, that's how they get the food. So I was like, grow the food for the city.
That's perfect. Yeah. So a chloroplast could be like a farm in a city. So some kind of food production area. Chloroplasts are essentially making energy or making food for a cell. That's perfect. I love it. Thank you guys. Okay, so mitochondria and chloroplasts are really important organelles in the cell. I guess all the organelles are really important, but I'm going to spend a little extra time talking about them right now. So first off, you want to know that mitochondria, here, I'm going to start sharing my screen with you guys so you can see it a little bit easier now, since I don't have to look at the breakout rooms anymore. Okay. Way down here. Okay. Here we go. So these are the energy centers of cells. Mitochondria are in plants and animals. So they're in pretty much every eukaryotic cell you can think of. You don't have to know what cellular respiration is right now, but just know that that's what produces ATP. You'll learn all about it in the next unit, I think, when we get into it. It makes ATP, which is energy. When you see ATP, think that's energy for the cell. So mitochondria in plants and animals, there's plants are the only ones that have chloroplasts. So chloroplasts are basically what allows for photosynthesis in plants. We don't photosynthesize. We can't take energy from the sun and make our own food. As awesome as that would be, it's not possible because we do not have chloroplasts. We only have mitochondria. So chloroplasts are, converting solar energy into chemical energy, which is kind of the energy that we use. So that's why we end up having to eat plants, because they're essentially taking that ultimate source of energy and making it available to us. An interesting aspect is that they actually have their own DNA. And I'll show a video on Monday about how this kind of evolved, basically how the first eukaryotic cell evolved from prokaryotic cells eating each other, more or less. But these are the only organelles that have their own DNA. Otherwise, the only DNA is in the nucleus. Sorry guys, I didn't do that. Okay, there we go. So the mitochondria, I'm just gonna go through a few characteristics. You guys will learn much more about the mitochondria when we get into cellular respiration, because you're gonna learn why all of these infoldings of the membrane happen. So it's this kind of crazy maze within a mitochondrion, but it's really important for the process of respiration. Don't worry too much about most of this information. This is just kind of extra, but I really want you to know mitochondria make the ATP of a cell for now. Like I said, we'll get into this. You'll know what all these pieces are in a bit. And then the chloroplast, kind of similar, but this is where photosynthesis happens. So light energy is going to come into here and be made into essentially glucose. So plants are going to make glucose or sugar by just taking in that light energy. Again, don't worry too much about the stacks and what all these things are called. We'll get into that, and I figure between now and then you'd probably not remember much of it anyway. So chloroplasts, that's where photosynthesis happens. Sunlight converted to chemical energy. That's the take home. And then, yeah, this is a video we'll watch on Monday. So we'll do that and then finish up the last couple slides and get into, I don't remember what next. Oh, the plasma membrane. Yeah. So that's it. And thank you for participating in that activity today, and have a good weekend.